member of the symbiosis team. I'm presenting the work on microbiology, composition of the human oral microflora depending on its diet. We have built several aims and purposes for this work. The main purpose of this work is to prove that people with a different diet have different oral microflora. But let's start by going deeper into the topic. The human oral cavity provides a unique ecological system for the most diverse microbes that create long-term microflora. The availability of food resources, constant humidity, pH, and temperature make suitable circumstances for the adhesion, colonization, and reproduction of all kinds of microbial appearance. The human nutrition menu has the ability to influence which bacteria and in what amount settle in the oral cavity. We have built goals and objectives. Surprisingly, approximately 160 types of microorganisms live in the mouth of a healthy person. You probably had to hear that the mouth is the dirtiest place in the body. This statement is partly true. The oral cavity is one of the most populated parts of the human body. Microorganisms enter the oral cavity with food and water as well as from the air. It is in the mouth that the most favorable conditions for the development of bacteria are observed. In this part of the body, there is always uniform humidity and temperature, approximately 37 degrees Celsius. Abundance of nutrients, sufficient oxygen content, presence of fluids in the oral cavity, internal spaces, and gingival pockets, slightly alkaline pH provoke reproduction of various bacteria. The oral microflora is extremely diverse, but it should be noted that a significant part of the oral microorganisms of adults are anaerobic species. Oral microflora is affected by diet. Unfortunately, modern person consume a lot of carbs and it causes caries. Bacteria use carbohydrates to nourish and build plaque matrix, and the end product of this process is organic acids, which cause the animal to demineralize. In our work, we decided to investigate the bacterial flora of oral cavity of dependence on the diet. The survey identified groups of people with the most expressed food preferences. As a result of the survey, 3 groups of school children with different food preferences, such as carbs, protein, or fatty foods, were identified. Samples of plaque were taken from the 3 school children of each group, which were placed in 1 milliliter of sterile water, after which microflora was sown on petri dishes with mid-heptone agar. The cups were placed in a thermostat at 30 degrees for 2 days. On the second day, the grown colonies were analyzed and microscoped. As you can see on the slide, we have 3 different colonies. These results are very interesting. At the end of the work, we made the following conclusions. First of all, plaque has a lot more bacteria than saliva. Second of all groups of school children had staphylococcus, and the main ones. Teenagers who prefer protein food have discovered staphylococci, fusobacteria, streptobacteria, and veillonella. Teenagers who prefer carbs have streptococci and staphylococcus. And finally, teenagers who prefer fatty foods have staphylococci, peptococci. And based on the work, we made the following tips and advice. First of all, everybody should have a variety of food in composition in order to keep the oral cavity healthy and diverse. Second of all, as you all probably know, brush your teeth twice a day. In the morning, after the breakfast, and in the evening before going to bed. And last but not least, visit a dentist at least once a year. That's all I want to tell you. Thank you for your attention.